Hey there, how's things going? Okay, we did, I'm back to chemistry, even though I know the cell membrane and diffusion and stuff is in between there. But we did inorganic chemistry, atoms, protons, neutrons, electrons, that kind of stuff, bonding capacity, valence, got it. And then we did biochem. Biochem was long, lasted about 2 hours and 1 minute. And I don't expect you to sit there for 2 hours and then take notes and stop it, spend 6 hours in front of the computer without going nuts. I got it. That's why I want you to stay ahead a little bit. So that one you could have ran it off half of it in an hour, straightened out your notes, went back, took maybe another 2 hours, and then the next day start fresh. You got a whole week. Got it? Anyway, we finished biochem, the macromolecules, proteins, carbohydrates, lipids, and nucleic acids. We saw how they're put together. We talked about pearl necklaces, what the pearls were. We talked about saturated fats, unsaturated fats, double bonds. We did some functional groups. We talked about what an amino acid is, and I kind of showed you that stuff. Yes? Okay. Now, jump down your syllabus. That's the one that has the light bars on it. I don't have mine with me, but I think you jump down to 1 week and you're going to see protein synthesis and cellular respiration. I'm going to do protein synthesis first, and then I'll do cellular respiration for you. Good? All right, here's what we're going to cover. Maybe. What's the importance of DNA in protein synthesis? I think one of the things that COVID has done for us as educated community is you understand what a virus is a little bit. You understand how a virus reproduces. We understand what our DNA is for. And we have this word that's come into our language called antibodies. Antibodies are proteins, hence protein synthesis. It's one of the things that's tied up in your DNA code. Your DNA code, we always think of it, oh, it's blue eyes, brown eyes, curly hair, color of your skin. It also, we've learned that it is making enzymes, insulin, hormones, insulin, glucagon, we talked about those things, the digestive enzymes, pepsin, and the list goes on, okay? I'll give you a quick insight to something you should know. I've been able to drink my milk my whole life, and all of a sudden I can't drink it. I get stomach problems and I have diarrhea and stuff. What has happened? You have lost the genetic code to produce an enzyme called lactase, ASE. Remember our thing? Substrate plus ASE is the enzyme. The substrate is lactose, which is just sugar, milk sugar. And lactase is the enzyme that does it. It's tied up in your genetic code. And for some reason, I don't know where it's at on the gene or what gene it's on, we have lost, some of us have lost the ability to recognize that. And when we do go through protein synthesis, specifically targeted for making lactase, we don't have a genetic code, or it's blocked for some reason. So therefore, we can't produce the enzyme. That means that disaccharide, lactose, it can't be absorbed, and it causes irritation to the digestive tract. And boy, do we know that, huh? So they put milk that lactose has been removed, and we can digest it. Okay, rice milk and coconut milk and all those other milks don't have it. Okay, fortunately, I love milk, especially ice cold, fresh, and I have the enzyme lactase. Anyway, the ability to make anything, any protein, is tied up in your genetic code, and that's what you got at birth, with mom and dad and the zygote, all that kind of stuff. Yes, can we lose that genetic code? Yes, we can. Can we find it again? And the answer is sometimes that works. We have these little guys cruising around in your cell that their job is to make sure your DNA code is correct. If it's not, they stop the cell doing what it's doing. They try and fix it or destroy the cell, called gatekeepers. Anyway, what is DNA? What's the importance of it? You got to have it. All of your cells, every one of your couple trillion cells, all in the nucleus have the same genetic code. All of them. It isn't like one has one and one has another. It has all the same. So your eye, theoretically, your eye cells can make finger cells, can make ear cells, can make heart cells. But that code has been turned off in that location. Right? Okay. How is the nucleus involved? Well, that's where we put our DNA. Got it? We had to put it somewhere. So we decided, I'm gonna have a little popcorn. We decided to put it in the nucleus. When we want to make a copy of a portion of that DNA, which we call a gene, what we do is we start the reproductive process. We call in a regulatory gene. There it is. That's what we want. And we copy that. And when we copy it, it's called something. We don't say copy it. We say transcription. We just transcribe that genetic code into a carrier that carries it out of the nucleus and over to a ribosome. That carrier is called messenger RNA, lowercase m RNA. Got it? That way you never lose your DNA. There it is. What did I just say? Transcription. Transcription is the making of what? Not the making of a gene. It's the copying of the gene. It's making messenger RNA. Popcorn's pretty good, you know? Especially if it's dripping with butter. Non-fattening butter. Okay, so you know what transcription is. You're going to see it on exam. The answer is the making of messenger RNA. What's a ribosome? A couple things about ribosome. I sure hope you're taking notes. Ribosome is the most common and numerous organelle in a cell. Most common and numerous organelle in a cell. It's outside in the cytoplasm. When messenger RNA is made, it exits through a hole in the nucleus called a nuclear pore, and it goes out to a ribosome. A ribosome is where protein synthesis occurs. Did you get that? There's a test question. Ribosome is where protein synthesis occurs. Digest that for a minute. This is called multitasking. So you know what a ribosome is. What is mRNA, rRNA, and tRNA? You know we talked about the first one. M stands for messenger, R stands for ribosomal, and T stands for transfer. Okay? Other than the ribosomal, if you just kind of put the ribosome, instead of saying ribosomal, you put reader, messenger carries the message, R, ribosomal, reads it, and T, transfers amino acids. Okay, you're gonna learn that. How are nitrogen bases and complementary base pairing involved? Remember that? A to T, G to C, except for in RNA, the T is not there, it's a U, and A recognizes U just like it recognizes T. That's how you hold the molecule together, and that's how you keep all your nucleotides in the right spot. Got it? And when they bond, they can only bond one way, and it's called complementary base pairing. The base are the nitrogen bases. We good? Okay, what are codons and anticodons? You could understand, it's common sense that if you just had an A or a T out there and it triggered for an amino acid to come in, then that'd be easy to get jacked up. So what your cell has done, which is pretty clever, is it's put combinations of 3 nitrogen bases together to signal for that amino acid. So an A doesn't do anything, a T doesn't do anything, a G and a C doesn't do anything, a U doesn't do anything. But if you put 3 of them together like an ATT, it'll trigger something to come over. You get it? Kind of like a fail-safe. That's a codon. That's a codon. Codons are only found on messenger RNA. And I'm giving you a lot of information. I hope your little hand is just moving quickly. Okay. If eat in front of you is rude, tell you what, go get something to eat and then we'll be even. So codons are on messenger RNA, anticodons are on transfer RNA, tRNA. Anticodons carry the amino acid. Codons carry the message. Codons and anticodons complementary base pair in order to deposit the amino acid. Whoa. What is translation? Well, remember the rRNA I said is the reader? So after you transcribe and make messenger RNA, it goes out of the nucleus through the nuclear pore to the ribosome. The ribosome doesn't read it. It's just the place where it happens. What reads it is rRNA, ribosomal RNA, and it reads codons. So every time 3 codons come around, it will tell, in the cytoplasm, it will tell transfer RNA, I need these 3 complementary base pair codons, and with it, I'll bring a specific amino acid. So when you tell me what you want, and I'll bring you the amino acid that goes in sequence. And this is our primary structure. Remember that? Primary structure was the amino acid sequence. There you go. Translation is reading and translating. I didn't want to use that word. Reading and translating the messenger RNA. Where do the amino acids used, that should be where do amino acids used, comma, come from? Where do they come from? I want you to think about this. Is it magic? Okay, I hope you're kind of giggling and go, well, of course it's not magic. And what is it? Where do they come from? Okay, I think I have this asked on your lab, or I used to. I had this on a lab, because I wanted you to think outside the box. Once you think about it and you get the answer going, well, that was obvious. I'm doing it right now. What am I doing? And it ain't eating popcorn, man. Okay, I'm eating. When you eat anything that has amino acids into it, your body's going to break down that pearl necklace into pearls, amino acids. It's going to travel in the blood to the cell that needs it. Pretty good, huh? What's a polypeptide is the same as a protein. Okay, remember what a monomer is, a monomer? Okay, and we gave examples. We said a monomer can be glucose, a monomer can be an amino acid, a monomer could be a nucleotide, huh? Remember that? Poly just means you have a whole bunch of polymers. A polypeptide means you're only talking about proteins. I have a whole bunch of amino acids strung together in an almost necklace. It is a polypeptide. Once it becomes functional, it's a protein. So there is a difference. Good? Okay, here we go. Transcription. Okay, the next topic is transcription. I finished my lunch, that popcorn. Watching my boys figure. Transcription. You realize I'm only 25 years old, don't you? I just had I've had a hard life. I've been bad 25 years. Okay. I think this is energy drinks caused all this. All right, transcription. Remember we said, what was the definition of transcription? The making of messenger RNA. Okay, here's a picture. Here's your DNA. It's been unzipped down here using an enzyme called helicase. You don't have to worry about that. And this is the making. The blue strip is copying this piece right here. This piece it's copying has enough nitrogen bases in it that we call it a gene, and it has enough coded information to make something. Okay, you see RNA? Just saying this is RNA, but this is actually messenger RNA, and this is going on in the nucleus of the cell. Once this is completely done, picture there's a big shield right here with some holes in it. This will sneak out the holes over into the cytoplasm onto a ribosome, where ribosomal RNA is waiting for it. Also out here in the cytoplasm is a transfer RNA with an amino acid on it, waiting to be called. Okay, now you back, and I said, here is your DNA. Here is your DNA. Don't worry about this. Here's your DNA. You see G to C, C to G, G to C, T to A to T. We know if you see a T, it's DNA. And here it is stretched out. So we unwind that double helix, and this is what we get. Okay, now it could be this section right here is a gene for something. It could be this section right here is a gene for something else. So what we do is we tie it in, and when they bond together like this, it's called complementary base pairing. 3 of these, so not the whole thing, just the bottom part in this case, because it's on the bottom, could be on the top. 1, 2, 3, 3 make a codon. Codon number 1, methionine. That is an amino acid. The next 3, 1, 2, 3, which is GGG. Nope, I'm sorry, GGC is codon number 2, and it's glycine, another one of the amino acids, and so on and so forth, all the way down, codon, codon, codon. Not one of these calls for amino acid. It takes 3, 3, 3, 3, 3. You get it? And those are codons, and codons are being read. This is messenger RNA. It's going to be translated at a ribosome by rRNA, and it's going to say, hey, transfer RNA. I need methionine, and I want you to bring me, and how it does it doesn't say, hey, on an intercom. What it says is, according to my complementary base pairing, my A needs a U. My U needs an A. My G needs a C. And this portion right here, which would be down here, you haven't seen it yet, I'm going to bring it in, would call for methionine. Wow. mRNA transcription takes place in the nucleus, making a copy of a gene with specific order of nitrogen bases, and that is called transcription. We already wrote that down. Yes? See another word, gene here, messenger RNA. They're tied in together. Messenger RNA then leaves the nucleus via nuclear pores, which is just a hole, and attaches to a ribosome. We already talked about that. A ribosome has a large and a small subunit. That's not that important. RNA is located here. That is important. That's all. What else do we say about ribosome? Come on, come on. Pause it. Look through. If you pause it, if you can't remember and you're thinking, pause it. If you remember, you have to look through your notes. Also, look through your notes so you know it's there. If it's not there, Jacko, you messed up. Okay, make sure it's there. Remember, it's the most common organelle in the cell. rRNA calls for specific anticodon. What happens is messenger comes out to a ribosome. As it's going through the ribosome, ribosomal RNA is translating it. It's reading it, translating it, reading it. Okay? Transcription is making it. Translation is reading it. And when it reads it in codons, 3 nitrogen bases. Can't read 1, can't read 2, it's got to read 3. It's got to be right. If it isn't right, it doesn't call in for squat. If it does read it right, it calls it, it says, hey, transfer RNA, complementary base pair, bring me the opposite of these things. Along with it, bring me your amino acid. If you don't have any amino acid, that person hasn't eaten any protein, don't bother coming, man. Okay? So we've said RNA calls for specific anticodon. It's on a transfer RNA, and a complementary base pairs with a codon. Codon, 3 nitrogen bases, connected as directed by your specific DNA. Wow. Did you get that? That's heavy, huh? Translation, the reading of messenger RNA by ribosomal RNA at the ribosome. The reading of messenger RNA by rRNA at the ribosome. Okay, we already talked about that. From translating messenger RNA, I'm giving you a little story. From translating messenger RNA, rRNA calls for specific anticodon, which carries a specific amino acid. Got it? If your complementary base pair is a GCU, it's only going to call in one specific amino acid. It might be lysine. Do you understand? Okay. tRNA has an amino acid on one end. This is important. It's a test question. tRNA has an amino acid on one end and an anticodon on another. Okay. Also has 3 nitrogen bases. obviously, because it has to match up, right? Okay. The complementary base pairs with a codon of a messenger RNA dropping off the amino acid, yielding a polypeptide eventually. First time, it's a mono peptide, then a dipeptide, then a tripeptide, and a quaternary peptide, on and on until you got an oligopeptide, which means a smaller than, and then you have a polypeptide, and then what, boop, you make that necklace active, it's now called a protein. Okay, now let me stop you here for a minute, because I think you're just looking at this PowerPoint, you're going, man, I have all this information here, I don't have to write down anything. You are so wrong. If you listen to me talk, I've been talking for 20 minutes, man. We've only gone through 3 slides. I'm giving you the story. I'm teaching you not just to memorize terms and what they mean. I'm teaching you how to memorize things so they make sense. It's a story, Jack. The story is, how do I make proteins? That's it. Well, there's got to be some people in there helping me out make them. Yes, they're messenger RNA, transfer RNA, your genetic code, yada, yada, yada. This polypeptide is not a functional protein. Polypeptide is not a functional protein. This only yields the primary sequence. Remember, primary, secondary, tertiary, and sometimes quaternary? It must form secondary, tertiary, in some cases quaternary. Remember, it's a string. Then it twists. We call that an alpha helix. And that's secondary structure, also called pleated sheet. And then we wind it back on itself. Remember how I did it with a cord? Winds it back on itself. That's our 3-dimensional shape, and that's tertiary. And then sometimes if it takes 2 or more amino acid sequences, then it's called quaternary structure. Got it? Okay. Okay, so we know what a polypeptide is, we know what a protein is, we know what primary sequence, secondary, and tertiary. Good? Good? Okay. Here's a picture. They say a picture is worth 1,000 words. I hope my words mean more than the pictures, but here we go. Over here was the nucleus of the cell. It had holes in it, and we copied a section of the DNA called a gene. We made a complementary base pair, which means opposite, okay? And we made a messenger RNA in a process called transcription, and this is the messenger RNA. And what they did for you here is they just said A, B, C, D. Those are codons because they're on messenger RNA, you see it? And those codons called in for specific amino acids like this. So picture this as a transfer RNA. This is an amino acid, and these are your 3 nitrogen bases that deal with complementary base pairing. So they said A brought 1, B brought 2, C brought 3, D brought 4, and it's right there. You see it? And it's dropped off. This is becoming a polypeptide, which hopefully eventually becomes a protein. Here's the last one that just came in, number 5. Okay, regardless of what these are right now, just know that there's 3 of them. This is a codon, this is an anticodon, and it's calling in for a specific amino acid. This is what it would look like if it was blown up. So here's number D, here is letter D, and it was on the messenger RNA, the code was CUA. On the transfer RNA, it was the opposite, GAU. And with that came whatever amino acid was number 4. Here's number 6 getting ready to come in. D, attaches, drops off number 6. Then it ends up going away empty, and it looks for more amino acids that you ate when you had dinner. Are we good? Just a minute. That says the same thing. It says the same thing. Are we good? That is protein synthesis, Jacko. Okay? I've explained to you why it goes on. I explained to you the different types of amino acids, proteins, antibodies, and structural protein, all that kind of stuff. I did that, and I showed how it was done. You start with transcription, you go to translation, you bring in the amino acid, you start all over again. When it's done, it shuts off, it dissolves, and you make it again later when you need it. Good? Okay. Next one, cellular respiration. All right, Cellular respiration, like protein synthesis. Now shut your brain off. We're done with protein synthesis. We're going to go to synthesis. We're going to go to an outline as to why do I need to make something called cellular respiration. Cellular in the cell, respiration is oxygen. Okay, I'm going to make ATP. Whenever you put a phosphate molecule onto anything, it's called phosphorylation. You understand? So if I put it on an ADP molecule, it will be called phosphorylation and produce an ATP molecule. Do you understand? If I took that phosphorus and put it on your sandwich, it would be called sandwich phosphorylation. Get it? Phosphorylation, to put a phosphate molecule on a specific molecule. Why do we need to do it? Phosphate is a freaky little element out there that has a whole lot of energy in it. Remember the bonding capacity 1, 2, 3, 4, 5, 1, 2, 3, 4, 3, 2, 1? Remember that? Made sense, huh? And when I said it's easier to accept 3 than try and gain 5, you got that. Phosphorus can do 5. It's a weirdo. So it can do 5. It can have a bonding capacity 5. Got it? Okay. What is anaerobic? Look at this. See the word aerobic just right here. Aerobic, aerobic. Ana and an mean without. Aerobic means with. Aerobic means with using oxygen. Anaerobic means without using oxygen. How to not use oxygen and have respiration? Well, it's called, in other words, called fermentation. Okay. Does our body do both? No. Our body only does aerobic. No, it doesn't. It does anaerobic too. It's very, very inefficient. I'm going to show you why. Where does this occur at in animals? I'm worried about plants. Plants is another beast. I can explain that to you. Okay, it's called photosynthesis. But right now we're worried about animals. Where does this occur at in the animals? Well, in the cell. Good. That was very good. What did I use the term? Just a minute. Grasshopper. Okay. What's the overall reaction? Okay. Remember the molecular formula? Remember it had the number of atoms on it? Structural formula is how it looks. Molecular formula is how many molecules of atoms are there, or how many atoms are there. The overall reaction. I want to know that, Daddy. What are the 3 metabolic pathways? You're going, I'm so tired. I want to do chemistry. I wanted to do anatomy and physiology. Well, bucko, you're doing physiology. That's what this is. In order to understand the physiology, I got to understand the basics of chemistry. Believe me, once we're done with cellular respiration, you're done with the hardcore chemistry. And this is not hardcore chemistry, okay? Just the basics. Metabolic pathways. It is a pathway. You start with something A, abbreviated A, and you end up with something Z. Got it? And the pathway you took, or remember I said, anytime you break something down or building up, you need an enzyme and may or may not need water. It's either going to give off water, which is reduction, which is anabolic, or it's going to take water to break it down, and that's catabolic. Got it? Okay. There's 3 metabolic pathways in cellular respiration. I'll give them to you right now, but I'm going to show it to you in just a minute. It's glycolysis, Krebs cycle, you've heard of that one, and electron transport, sometimes called respiratory chain. What is NAD and FAD? You're going, oh my God, I'm so tired of learning all these terms. I'm making you smarter, man. Okay? NAD and FAD are coenzymes. Okay, they're a protein. Now, remember we had proteins that were enzymes? We also have proteins where we have to add something to it in order to make it active, make it a protein. Otherwise, it's just a polypeptide chain. What do we add to it, Daddy? We add, in some cases, we add an organic molecule like a vitamin. Okay? Nicotinamide is a vitamin. Okay? If we add that to a polypeptide chain, we form a compound called NAD, nicotinamide adenine dinucleotide. FAD is flavin adenine dinucleotide. It's a different one. So we have two types. Well, we actually have three. We have a protein enzyme, which is just amino acid sequence, primary, secondary, and tertiary. It's a protein. There's another type of protein that we have to add a vitamin to, which is called an organic molecule, and that is called a coenzyme. If we add an inorganic molecule, like the things on the periodic chart, I'll give you an example. If you add iron to a protein, you get a cofactor. And the example of an iron added to a protein is hemoglobin. Globin is the protein, heme is the iron. You put them together, you have a cofactor. You get it? These are not cofactors, these are coenzymes, and they're very important in production of ATP. What are the waste products produced and where do they go? Well, whenever you have a reaction, you know that you're getting waste products, products that you don't need. In the cellular respiration, the waste product is going to be carbon dioxide and hydrogen. Hydrogen eventually will bond with the oxygen that you breathe in, like that, and form water. Carbon dioxide, you expel in your lungs. Whoa, that's cool. Does it always begin with glucose? Keep talking about glucose, glucose, glucose. Nope. Nope. The thing is you're saying, well, why did we start with glucose? Because glucose starts at the very, 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 very beginning. So it's step 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. If we start with a lipid, we might start with number 11. If we start with a protein, we may start with number 17. So it becomes less efficient. So if I want to show you the whole picture of glucose, I'll start, the whole picture of cellular respiration will start with glucose because you can see every step. Okay, phosphorylation means putting phosphorus, P, atom on a molecule. And there are different energy forms that this can happen with. Okay, you understand? Test question. Phosphorylation is what? Putting phosphorus on a molecule. I don't care what it is. Got it? Phosphorylation, using the sun's energy. No, I'm sorry. Let me back up. I'm going to back up. There are different types of phosphorylation. The phosphorylation means I take energy. It takes energy to put that phosphate on that molecule. If I'm putting phosphate on this, and here's the phosphate, do it this way. You saw me, I had to put my hand over there. That took energy. Got it? It isn't magic. It is, oh, you like each other? Hey, go hook up. It don't work that way. What you say is, I have to have some source of energy. That source of energy can be the sun. It can be, just a minute. It can be the sun. You're cracking me up. It can be the sun. It can be a substrate. It can be oxygen. So if it's the sun, we put photophosphorylation. If it's a substrate like glucose, we put substrate phosphorylation. That's how the phosphate got on there. If it's oxygen, which is the end product of electron transport, it's called oxidative phosphorylation. You get it? Okay. Photophosphorylation uses the sun's energy. Next, chemophosphorylation. I didn't say this one. It uses chemicals. Okay? Sun, you know, is going to be photons. That's pretty straightforward. Substrate's going to be hydrogen and stuff like that, and transferring energy and stuff through those electrons. I get that. What was the other one? Oxidative means I'm going to use oxygen to get this thing going. Chemo means I'm going to use chemicals. Do animals use chemophosphorylation? No. What uses that? Mostly bacteria. Got it? Okay. Substrate, you break down. That did not go well. Try that again. I hate it when it gets ahead of me, man. This is a smart computer. It says you guys are way ahead of me. Substrate phosphorylation, that's when we use glucose. Glucose is a substrate. We're going to do something with it. We're going to manipulate it with enzymes, and we're going to get ATP. The last one is oxidative, using oxygen. This is the one we find in us, okay? We find two of them in us, substrate and oxidative. You good? Okay, aerobic versus anaerobic. Aerobic means you need oxygen, okay? In a normal cell doing a normal thing, it's aerobic respiration, oxygen, and we need oxygen. And it's going to form water. That oxygen's down there going, come to me, come to me. I'm very stable. If you attach to me, hydrogen, pay attention. If you attach to me, we can make a very stable compound that we can either recycle or we can urinate out, and that's called water. Very strong acceptor of hydrogen. Aerobic, that means without oxygen. It says in this context, means there is a lack of oxygen present, but a smaller amount of ATP is still produced, sometimes referred to as anaerobic respiration, through a respiration on there, or lactic acid fermentation. I use the word fermentation. You say, oh, my muscles are killing me because I just got up off the chair and went to the refrigerator. I am so tired, my muscles are killing me. What? Why is your muscles killing you? Well, because the cells are filled up with lactic acid. You got that right. You're correct. Lactic acid is a product of anaerobic respiration. And I'm going to explain that to you. Okay. Let me see what this says. I got to drag this down so I can see what it says. Overall reaction. Remember we talked about that? Okay. That did not go well. Okay, now this is a stupid drawing. I don't like them. So what I did is I showed you what's in your book. I'll explain what's in your book. And then I've actually physically drawn them for you. I think that'll make more sense, I think. Okay, here's ADP. ADP is still a source of energy, but we don't use that one very often. We use ATP. D stands for di, T stands for tri. Inorganic phosphate. If we take ADP, add phosphate to it, phosphorylation, we turn it into ATP. See it? 1, 2, 3. That means a high energy bond. That little wavy thing means a high energy bond. Got it? Here's the overall reaction. C6H12O6. What is that? Come on. Come on. I've told you this thing so many times. I almost want to reach through this thing and slap you. You're not thinking. You have to think. I've given you this information. Not only do you have to, once you understand it, you're going, this is like a little story. I get it. Then you put the terms with it. Then when I ask you this on an exam, you're going, hey, baby, I got this nailed. Okay, glucose, C6H12O6. Remember that? CH2O parenthesis N. You remember that? Okay, plus oxygen. So you take in food, sugar. You add oxygen to it. Okay, this is what we do when we eat and breathe. And that gives us 6 carbon dioxide. We blow that out. And 12 waters we can either recycle or urinate out. And look at this, 36 of 38 ATPs. OMG. From these two things right here, I got that much ATP. I can build a house with that much ATP. No, you can't. No, you can't. Okay. Okay, 3 metabolic pathways. Remember we talked about them 3 metabolic pathways? Remember that? Remember that? I said a metabolic pathways, you start with an A, you get down to a Z, and there's enzymes that come in between there. They yield something that we want. The 3 metabolic pathways are glycolysis. This is a test question. Metabolic pathway number 1 begins with glucose. It either ends in pyruvic acid aerobically or lactic acid anaerobically. Do you understand? Hold on. I keep pushing the goddamn wrong buttons. Cytoplasm. The first step occurs in the cytoplasm of the cell. Okay. So what happens is, you've heard of the mitochondria, and you probably learned this from high school or something. Says, what is the mitochondria? Oh, teacher, it's the powerhouse of the cell. Oh, good, Johnny, you're so sharp. I'm going to teach you much more than that, man. Yes, does most of the energy come out of the mitochondria? Yeah, but the first step, glycolysis, comes out of the cytoplasm. Good. Okay, remember that NAD, which I said was a coenzyme, stands for nicotinamide adenine dinucleotide? NAD. Okay, it's a coenzyme. This is a coenzyme that has an extreme affinity. That means it really likes it, baby, for hydrogen. When it's released from the catabolic breakdown of glucose, it reaches out and snatches it up. So when you take glucose and you bombard it with water and an enzyme, hydrogen comes off. Hydrogen's go, man, what do you want to do with me? And it says, NAD says, come to me, Jacko. You come to me, I will turn you into a lot of energy. Got it? NAD. ATP, oh no, I'm sorry, ADP, adenosine diphosphate. This is in the cytoplasm along with that. See that lowercase I means inorganic phosphate. The energy released is enough to cause phosphorylation. Okay. ADP can give you energy. ATP gives you more energy. Which one are you going to pick? Okay. So in our bodies, can we use ADP to give us energy to do something? The answer is, yeah, we got it. Problem is, if you take just a little bit more time, throw another phosphate on there, phosphorylation, you get ATP, you produce one heck of a lot of energy, and it comes very easily. Okay, now pictures. Remember, pictures worth 1,000 words? This is, I'm going to show you, let me see, make sure. Well, it doesn't want to pay attention to me, man. Okay, we have anaerobic, aerobic versus anaerobic, glycolysis, and I'm going to give you a picture. Okay, now remember I said I wrote them myself? Okay, I tried to simplify it. Your book makes it, I think, makes it difficult. I had to learn it that way. And I thought, well, why make it difficult? Why can't we just make this straightforward? Pathway, metabolic pathway number 1 called glycolysis. It starts with glucose with a 6-carbon compound. Remember C6H12O6? This represents a metabolic pathway. There's 7 steps in here. I'm just showing you 2 of them. So you take 2 NADs, nicotinamide adenine dinucleotide, and because of the breakdown of glucose, you're going to put out 2 hydrogens. Remember, the hydrogens are going, hey man, what do you want from me? NAD says, come here, bud, come here. We're going to form a bond. We're going to be buddies, and it forms NADH. So you're going, well, what the hell is NADH? That NADH is a way of transporting hydrogen through the plasma membrane of the cell. And once it gets to electron transport, it's going to drop off that hydrogen and produce buckets of energy. Also, we take ADP plus 2 phosphates, 2 of them, there's 2 of them, okay? So one of them is ADP plus 1 phosphate, another one is 2, and I just clumped them together. Gives you ATP, and you're going, well, shoot, that's all I wanted right there. Good, but you can only do a little bit of it. When you break down glucose using enzymes, you're only going to get 2 ATP. They ain't enough for you to even blink, Jack. Okay, so you're going to need these guys to work later in the mitochondria to produce a bunch more. And the result is from the breakdown, remember I said A to Z? The A is glucose, the Z is pyruvic acid, which is a 3-carbon compound. There's 2 of them, so you still have your 6 carbons. Do you see? So you haven't really lost anything but a bunch of hydrogens. This occurs in the cytoplasm. Under anaerobic conditions, you've got to dump off them hydrogens. This has hydrogens in it. Okay, you've got to have that right there. And when this is in this condition, you can't have that. You got this. So you got to dump off that hydrogen. If you dump that hydrogen off to pyruvic acid, so that hydrogen goes to pyruvic acid, you'll produce lactic acid and NAD. Then that NAD can go back up here and you can get 2 more ATPs out of it. So you get 2, 2, 2, 2 with every glucose molecule. Remember, you want 36, 38. So you have pyruvic acid, and the only way that you can get rid of that hydrogen is either have oxygen in place or dump it off to another, dump that hydrogen off to pyruvic acid, gives you lactic acid. And you know, with lactic acid, after a while, you can start getting pain in your muscle. Pretty soon your muscle won't work, huh? Okay, transition reaction. I don't know why this isn't working. trying to do something here. All right. Now let me do any of them more, so I'll just keep going. Transition reaction, two things occurring. It's a reaction. It's not a metabolic pathway. So it's a simple conversion, A to Z, or A to B, however you want to look at it. Okay? Pyruvic acid is being catabolized, broken down, to a 2-carbon molecule. Remember, pyruvic acid is a 3-carbon molecule. Remember that? Well, this is going to be a 2-carbon compound because a 3-carbon compound won't go into mitochondria. The passageways are too narrow. Got it? It's got to be this big. It's got to be this big. Can't be this big. So what you got to do is you got to trim off a carbon. When you trim off a carbon, you now have a 2-carbon compound called acetyl-CoA. CoA stands for coenzyme A. Remember coenzyme? A vitamin attached to a protein. Okay? Or a vitamin attached to a polypeptide to make a protein. It's 2-carbon. That means you've lost 1 carbon. Guess what you lost the carbon in? Carbon dioxide. Yeah. Acetyl-CoA is small enough to move into the mitochondria. So the first step, glycolysis, occurs in the cytoplasm. The second two, which is Krebs cycle electron transport, occur in the mitochondria, which you learned correctly as the powerhouse of the cell. 2 CO2s are released, and NAD are formed. Okay? So back in the transition reaction, you dump off the 2 CO2s, you know why, because it's got to get in there, but you also produce 2 more NADHs. NADHs are like ice cream in a freezer, baby. That means you're going to get something from it and you're going to like it. And what you're going to get is a bunch of ATP. So anytime you can either get straight ATP, as we did in the glycolysis, or we can produce NADHs, we want that. Remember, it's not a metabolic pathway. It marks the beginning of the Krebs cycle. Good? Good? Here's what it would look like. Now, what I did is I'm adding to our drawing. You remember this one, okay? This is aerobic pyruvic acid. Pyruvic acid, you dump off a CO2 and you get acetyl-CoA. 2-carbon, 3-carbon. There's 2 of them. So you got 4 carbons versus 6 carbons, which is what you had up here, okay? This gives you acetyl-CoA that goes into the Krebs cycle. But also when you broke down pyruvic acid, it kicked out a hydrogen. It says, hey man, get out of my life. You're going, oh, hydrogen goes, what? You're hurting my feelings. It says, okay, jump onto an NAD, because there was NAD there too. It says, oh, NADH. I like NADH because it's going to turn me into a lot of ATP. This squiggly lines, I meant to make those squiggly lines, the mitochondrial membrane. Mitochondria is like a circle out here, like a hot tamale. Okay, now we go to Krebs. You've always heard of the Krebs cycle, yeah? Going, oh, I know what the Krebs cycle is. Okay, good, good. I'm glad you do. But the Krebs cycle is just a minute. I'm trying something here. It just doesn't want to get rid of me. I'm trying. Well, I just want to do. Don't want to do nothing. Look at that. Okay. Anyway, Krebs cycle has 3 names. Krebs. Okay. Krebs cycle. TCA. That stands for tricarboxylic acid. Tricarboxylic acid. Now you know I'm going to ask you these on the exam. Okay. And also called the acid cycle. So those 3, which is metabolic process number 2, begins with acetyl-CoA. It's a 2-carbon compound. Okay. It says completely whoops. When you take acetyl-CoA, it'll give you 3 more NADHs. Don't worry about them NADHs that came out of glycolysis. Don't worry about those. I'll keep track of them for you. Don't worry about the NADHs, the 2 of them that came out of transition reaction. I'll keep track of those for you. Now we're going to talk about the ones that come out of the Krebs cycle. OMG, is there a bunch of them? You get for every acetyl-CoA, and we put 2 of them in there. So let's just look at one of them. It gives you 3 NADHs, and there's 2 of them. FADH2. Now we talked about FAD. We said FAD stands for flavin adenine dinucleotide. And it has 2 hydrogens attached. Yes, normally occurring in the cell, there's only 1. It's FADH. That don't do anything. It takes this hydrogen, adds it to it, and you get an FADH2. And there's 2 of them. okay? Because there's two from here, two from here. You have one raw ATP times in the Krebs cycle. We're talking about products of the Krebs cycle. Let me make sure you understand that. We're talking about what do I get from acetyl-CoA in the Krebs cycle? After it goes through this metabolic breakdown and all that kind of stuff, what do I get? I get an NAD, three of them. I get FADH2, I get two of them. I get one raw ATP and two CO2s for every acetyl-CoA. But I had two acetyl-CoAs from every glucose molecule. Got it? So we got to double everything. All right. Here's my picture. Whoops, didn't do too good a job there. Of the Krebs cycle, citric acid cycle, tricarboxylic acid, TCA. Tricarboxylic acid cycle, TCA. You start with two acetyl-CoAs, which are two carbon compounds. That makes 4 carbons. Remember the other 2 we lost in transition reaction as a CO2. Right in the very beginning, this is where it starts. You kick out another CO2. We're just looking at one of these, not 2, 1. You get a CO2. You throw in an NAD plus H and you get an NADH. So this kicks out an H, attaches to NADH, NAD and gives you an NADH. Remember, that's our buddy because it's going to give us a lot of energy. Right after that, we kick out another carbon dioxide. Now we had, from here, we had 1 left. From here, we have goose egg, nothing. We get another NAD. Wow, I like this NAD. Then we put in an ATP molecule. There's enough energy for phosphorylation to put that P onto a D, make it a T. So we got an ATP. Whoa, we wanted that. Then we take an FAD, and I didn't do this right. It should have been FADH plus an H gave an FADH2. Got it? This shouldn't be it. There's no free FADs. It's FADH. I messed that up. I'll go stand in the corner. And then after you do that, you get another one for NAD to NADH, adding the hydrogen. You understand? So for every acetyl-CoA, not 2, every acetyl-CoA, 3 NADHs, 1 FADH2, 1 ATP, 2 CO2s, 1, 2, those are waste product for every acetyl-CoA. This occurs in the mitochondria. You're going, Daddy, I'm getting this. Now we have to go to electron transport. Metabolic pathway number 3 begins with NADH or FADH2. Okay? As hydrogen passes down the staircase, and I'll show you what I mean with a staircase, in the form of electrons. Hydrogen, electrons, same thing. Remember electron whizzing around the nucleus? Hydrogen is just because, remember, it doesn't have any neutrons, just protons and electrons. So when we move hydrogen, we move electrons also. And then we can control them in the form of electrons. This is enough energy to cause phosphorylation, inorganic phosphorus, onto an ADP molecule, which gives us an ATP. For every NADH, you can get 3 ATPs. Remember in glycolysis, we broke down glucose 7 times and we got 2 ATPs. In the Krebs cycle, we broke down acetyl-CoA and got 1. Here, we take NADH, and just by transferring that hydrogen right there, we can get 3 ATPs. OMG. For every FADH, you get 2. That ain't bad. Not as good as FADH, but not bad. Cytochromes. These are another, when you see the word cytochromes, it's another type of coenzyme. And they don't have, it's actually coenzyme, cytochrome A, B, C, D, E. And they're not as strong of a bond as NADH or FADH2. So it takes, you have to release it, A to B, B to C, C to oxygen in order to get enough energy to make an ATP. So it says it's another coenzyme that are not as efficient as NAD or FAD. And it takes 3 steps, cytochrome A to C, to yield enough energy to make an ATP. Got it? You're saying, Daddy, give me a picture. All right. The final acceptor, here's a test question. Final acceptor of oxygen. The final acceptor of hydrogen. Now remember, NAD accepted hydrogen, FADH accepted hydrogen, pyruvic acid accepted hydrogen, turned into lactic acid. in the normal way of us breathing and eating and stuff like that, the final acceptor of hydrogen is oxygen. And it is very, very efficient. It's called oxidative phosphorylation, and it produces a whole bunch of ATP. Okay, I'm making a staircase. Do you see the staircase? At the top is NADH. I put a circle around the hydrogen because it tumbles down here. When it tumbles around here, you got an NAD and it goes back and picks up another hydrogen. This is FADH. It picks up that hydrogen, makes it FADH2. And then it tumbles down cytochrome A, B, C. And then it's enough to make another phosphorylation. So from NADH to FADH2, we have enough for phosphorylation, giving us an ATP. From FADH2 to cytochrome A, enough to give us an ATP. This one goes 1, 2, 3, and it's the last one to give it ATP. So if we start with NADH, we get 1, 2, 3 ATPs. If we start with FADH2, we get 1, 2 ATPs. And then we get a bunch of hydrogen down here saying, please help me, I'm drowning. Oxygen says, I am your savior. And it reaches over, grabs 2 of those and makes H2O. Let's look at some of the other stuff I wrote. FAD and FADH2 and cytochromes are all coenzymes. It says electrons are being transported as hydrogen. For every NADH, we get 3 ATPs. For every FADH2, we get 2. Final acceptor of hydrogen is oxygen, forming H2O water. Make sure you know everything on this because there's about 4 test questions coming at you. Well, I don't know what that says. Summary, I know what it is. Summary, I'm going to put this into a table for you. You're going to be very happy with me. Okay. What I did, I even typed this one up. We have NADH, FADH2, ATP, and CO2. CO2 is just a waste product, but I want you to account for them. Okay, in glycolysis, how many NADHs did we produce? Remember, it was on the right-hand side. I have a hook. We produced 2. How many FADHs do we produce in glycolysis? This is in cytoplasm, 0. How many ATPs did we produce? 2. How many CO2s did we produce? 0. Then we went to the transition reaction. So it was down, diagonal, transition reaction, we got 2 more NADHs. How many FADH2s? 0. How many ATPs? 0. How many CO2s? Remember, we had to dump off 1 for every pyruvic acid, giving us acetyl-CoA. So we had dumped off 2 of them. Now in the Krebs cycle, we produced, remember, for every acetyl-CoA, we produced 3 NADHs. And we did 2 of them. Remember, there's 2 acetyl-CoAs, gave us 6. FADH2, 1. 2 acetyl-CoAs, we got 2. ATPs, raw ATPs, we got 2 of them for every acetyl-CoA. Wait a minute. We got 1 of them for every acetyl-CoA. I got this sucker backward. That 1 should be over here. We get 1. Since there's 2 acetyl-CoAs, we get a total of 2. This is a total. And then for CO2s, just so we know, we get 2 out of Krebs cycle. Since there's 2 acetyl-CoAs, we get 4. We got 2 of them from the transition reaction, 4 of them from Krebs cycle. There's a total of 6. Remember our overall reaction, 6 CO2. NADHs, 10, 4, 4. So now we're doing a total. However, we're trying to get to that 36, 38 ATP. NADHs, you get, for every NADH, you get 3 ATPs. 3 times 10 is 30. 10, see them? 2, 6 is 10. 10 times 3, 10 times 3 gives us 30. I put it right here, 30 ATPs. FADH2, 0, 0, 2. Okay, since we get 2 ATPs from it, we have 4. So now we put that 4 down here. 2 times 2 is 4. We have raw ATP. We got 4. Okay, so we got 4. 2 for every glucose molecule. It ain't what I wanted. We got a total of 4. Here's ATP. We got 2. And so for every acetyl-CoA, Since there's 2 acetyl-CoAs, we get 4. That's at 4. 4 ATPs, 4 ATPs. You add this up, you got 38. You understand? You understand? Okay. Okay. Got 1 from glucose. Okay, it says other food sources that yield ATP. Fat. Remember that fatty acid chain that went out, that hydrocarbon chain? For every 2 of those, every 2 carbons, hydrocarbons, is acetyl-CoA. So every time you throw in acetyl-CoA, you just saw you got 36 ATP. So that's huge. So every 2 little carbons you get, well, you don't get 36, you get half of that. So you get 18, right? So 18, 2 of them 36. That's huge. So 4 carbon compound of hydrocarbons, you get 36 ATPs. Whoa. 10 acetyl-CoAs forms 20 carbons, gives you 100 ATPs. Oh my God, why do we store fat? There it is. Proteins either enter the Krebs cycle as intermediates, and although yield ATP is not as much. There was a guy that came out with oh Jesus, it's been 20 years, 30 years. Guy name was Atkins, Dr. Atkins. And he came up with the famous Atkins diet. He says, you can, on my diet, you can eat as much fat and protein as you want. Gorge yourself. Remember the 4 macromolecules? He's not working about nucleic acid. When he wants you to eliminate is carbohydrates. If you eliminate carbohydrates, you can only metabolize proteins and lipids. So you're either going to get acetyl-CoA, which gives you plenty of energy, or you have to work hard at it with proteins. Proteins doesn't give you many ATPs. Man, maybe gives you like 10, 12, 8. So that means you're eating and you're getting that satisfaction of, oh, I feel full, I feel really good. You're getting that satisfaction, but you're losing weight because your body's burning up fat. Make sense? He died, slipped on some ice in New York. Nucleic acid. I don't know what happened. Nucleic acids, they also enter the Krebs cycle. They don't even give you as much ATP as proteins. So we could, if you eat nothing but nucleic acid, which means when you eat a cell of an animal or a plant, you're eating that plant's DNA. We use that DNA, or we can metabolize it, make ATP. That's it, boys and girls. It's going to, now, I hit you with a whole bunch of terms. It's going to take you time. I get it. Take that time. Okay? Any questions? Get a hold of me, get a hold of Brooke, get a hold of Sarah. I really don't know what's going on with this thing. Hang with me. I may have to do something drastic. Stop video. Let's try that one. You know, my thing's not working. So I may abruptly unplug this because I'm going pause, share, stop, share. It's not allowing me to do anything. Okay, we're going to go to drastic measures. Whoop, something happened. I don't know what happened, but it looks like it might be catching up with me. Well, I can't pause it. You guys can check out here. This is my problem here. From beginning, hide, sec, stop share. You are sharing. Yes, I am. I want to stop share. Well, All right, let's see if I can change hotspots and that will help. Well, I'm not getting anything out of here. We're gonna try changing this. Still not letting me do anything. I don't know what to do. Um, this is not good. Alt Q. Alt Q. Alt Q. Let's see. Hi, darling.
Hi, handsome.
What you doing?
Going to the bathroom at Save Mart.
What are you doing in Save Mart?
Going to the bathroom.
That's it?
Well, no, I needed green onions.
Oh, that's right.
Yeah,
Okay,
you
you're
need
going to the
anything?
bathroom a lot.
I drank all my coffee.
Oh, I see.
Uh-huh.
Okay. I can't think of anything else we need.
Okay.
Okay,
All right, I'm almost home.
good. I can't wait.
See, there was a little bit of rain when I was outside of San Andreas.
Well, there ain't squat here.
Okay.
Okay,
All right.
I love you.
I love you, handsome.
Bye.
Bye.
Control. Control Alt C. Alt C.